Hi, Anna.
Hi, Liz.
It's great to see you again. Um, we're just coming to the end of your residency for Fabrica Algologies, which is co-commissioned by Fabrica and the Living Coast. And we're just going to have a quick chat, a bit of a reflection about how you find the residency.
Great.
Okay. I'm going to ask you a few questions, just as little prompts, but really it's all about you.
Always.
So, when you were selected for the residency, you submitted a really fulsome, a very actually sophisticated proposal for the residency, which is a very short residency. It's only a few weeks. And I think one of the things that I was a bit nervous about actually was how you were going to research all the different themes that you had submitted as part of your proposal, which I think the starting point for it was lockdown itself.
Mm. So, I mean, this is how it emerged. Was I was here during the lockdown and over the past sort of year and a bit now, and I was walking every day on the Undercliff and on the beach there, and I started to notice. I think I worked it out because there's a set of photographs, and it was August, so I'd already been walking on the beach, from March to August, and at that point I was like, oh, seaweed, it's actually kind of interesting that's doing this stuff, and then and changing it didn't look like this before when we were here, and it looked really odd. In fact, there was this red weed everywhere, covering all the pebbles, and I started to notice it then. And I talked to one of the interviewees on the blog off camera, said to me, he said, well, Anna, I know a grass researcher, and he said to me once that if you imprison someone for long enough, they'll start to find anything interesting. And I think that this is the situation with me and seaweed. And actually, I mean, it's a fascinating subject once you start looking at it. And then I started to find out all these connections, because I mean, that's kind of the way I see the world, is through all these interconnections, and how they're all linked and things. And I started to think about, well, I found out about Mary Philadelphia Merrifield and seaweed collecting in Brighton. And one of the sort of the most foremost seaweed collectors, these Victorian seaweed collectors, which were mostly predominantly women, was here in Brighton, and she wrote a book called something like The Natural History of the Flora and Fauna of the Brighton and Hove and its vicinity, or something like that, and collected a lot of seaweed, which is apparently in the Booth Museum and the Natural History Museum. So she identified loads. And she was also a fashion historian before that. And then she wrote this book and got really obsessed with seaweed and sort of dedicated the rest of her life to that. And that's really interesting to me, because I'm really interested in not fashion, but history of fashion as an indicator of time. So I have a lot of works that reference fashions of certain periods. And so that sort of linked to me as well, made another connection. And then, well, I was thinking about, well, a lot of my work is to do, I mean, most of my work is to do with microbiology. And I love the history of microbiology and looking at the future of microbiology. And an agar jelly, which is used in every single lab around the world, is made from seaweed. And the reason that we use it is because Fanny Hesse in Robert Koch's lab in Germany, in sort of the 1880s or something, he was trying to culture bacteria on gelatin. But when you heat it up to like human body temperature, it melts. And so the bacteria can't grow, it sort of gets sucked into it. And so they were having terrible trouble with this, and Walter Hess, who was the lab tech, was a member of Robert Koch's lab as well, and so Fanny Hess was his sort of unofficial lab technician. He said to her, well, how come your jellies don't melt like all these other ones? And she said, well, she was American, she was from New Jersey, but apparently she'd been exposed to people from lots of different countries and things like that, and so she'd started using agar jelly, which is a kind of Chinese dessert, to make her dessert. So she was using this, and she said, well, I use this. And I mean, she knew about the problems they were having in the lab as well. So I think, it's how the story's told. I'm not how sure, how, you know, whether her contribution, which sometimes these accidental contributions in science, like Alexander Fleming's somewhat accidental contribution to science, is discovering penicillin, which was actually produced by other scientists. But so she said, why don't you use this? And that's what they did. And every lab in every country in the world uses this. So then I started thinking about the history of women in science and the credit around that, and sort of through the lens of seaweed, as they all seem to interconnect them. So there was women in science, and there was this history of agar and history of microbiology. So I brought in things like microscopy and did like microscopic images as well. And also Anna Atkins is another of these. In fact, she predates, her publication anyway predates Mary Philadelphia Merrifield in Brighton, but Anna Atkins used cyanotypes and produced the first ever book of photographs, which is this book of seaweed, Cyanotypes. And it was the first photographic book ever published in the world. And it was seaweed again. So it's this interconnection. Then, just to conclude all these interconnections, cyanotyping is an old-fashioned technique and it's still very much used, and people are very fascinated in it. But a more modern but seemingly older technique, because it looks like something we should have been around forever, but it hasn't been, is this idea of botanical printing, where you can use leaves and plants and flowers and even seaweed in my case, and you wrap it very tightly into bundles and steam cloth with the leaves after you've moored into it. You have to do some chemical processes, and you can get the actual prints onto the cloth. And that seems very linked as well. So I was doing quite a bit of botanical printing, which are also on the blog now. And there's even a handy step-by-step guide to a basic method for botanical printing for people who are interested in that kind of thing. It is a really fun activity. It's a little bit fiddly, but I've simplified it quite a lot in the description, so people can try that. So I just basically got stuck in, I think, is the way, and started seeing all these fascinating connections. And you know, it just sort of, the art kind of makes itself, like the microscopic images, like the series of those, they just sort of made themselves as soon as I kind of started trying to look down the microscope at the seaweed. It's just so beautiful like that. But it is also disgusting. Some of it produces so much slime. It's like I've got a kind of, I went through a phase of having like bowls of seaweed, wet seaweed all around the house. And some of them are just like, ooh. It's really off-putting. So it's funny how, and it's obviously, it smells quite intriguing when you're making a botanical print out of seaweed, not like when you make one from a rose, for instance, which has a sort of nice rosy smell. You get a sort of weird seaweed smell. And of course, I made my own agar. Well, I tried to anyway. So that was another aspect of it, which took me on a whole journey.
That's brilliant. So we should remind people that they can discover all your experiments and some really deep insights into the research that you've done through the blog. It's a great blog.
Thank you.
I want to just draw a connection back to the Living Coast now. So the Living Coast, which is Brighton and Hove and Lewis Downs Biosphere. So the Living Coast is the kind of brand for the biosphere. There's lots of environmental improvement projects that the Living Coast is managing. Some of which are about the kelp beds along the coasts between Brighton and Worthing, I think. And I just wondered if you could just talk a little bit about kind of what you looked into in terms of seaweed and in relation to the Living Coast.
Well, one thing that was very interesting was a seaweed that sort of was quite a beautiful seaweed that kind of appeared out of nowhere at one point, and I was like, what's this one? And then there was a little bit of it, and I talked to Sarah Stewart, who's the phycologist that I interview on the blog, because I should say that algology is a misnomer, because back in the Victorian period when it was so named, people just basically thought you take a word and you put ology on the end and it means the study of. So they took algae and put ology on it, and so came up with algology. But actually that means the study of pain, so it's the medical study of pain, whereas algology, as we're using it, is actually phycology, which is from the Greek phycos. And so I got a phycologist to look at this, and she was like, whoa, that's Sargassum muticum. That's an invasive species, and it grows really fast. And so we have it down here now. So, and actually you can make things like alginate from it as well. So it's like, I was wondering about like the harvesting and conversations like that. So I discovered this, and there's some really beautiful seaweeds as well, these kind of red thread-like ones. We've got quite a lot of Irish moss as well, which is another lovely one. But the kelp, so I interviewed Ella Garrod, who's the Living Seas officer of Sussex Wildlife Trust, as part of the residency, and so the video's on the blog. And she was telling me all about the kelp rewilding. And so this idea, so it was things like trawling along the sort of kelp beds, but the main, so where it started was the great storm of, was it 1987? I think it was
Something
about
like that.
1987, yeah. The what we know as the Great Storm for those that were there, where we actually had a sort of proper hurricane in the southeast, and trees, most of like the trees on the level, well, it was quite tree filled, wasn't it, before? And so there was quite a lot of trees were felled in it, but it also affected the kelp forests and caused a lot of damage to that. So when that was more sparse, apparently trawlers that couldn't get there before were able to kind of trawl that area, and that sort of stopped the kelp from growing back again. And so what they're trying to do now is they've passed a kind of a law, a rule that the trawling's not, there's a sort of trawling exclusion zone, so they can't now trawl in that part, and they're trying to regrow the kelp in various ways, and apparently it's coming back. And kelp's good because in some parts of the world it is very good kind of carbon sink. It takes the carbon out of the atmosphere and stores it, but apparently it dies off and then it regrows, and the died off kelp needs to kind of get sort of sucked into the earth and kind of kept there. But they're not sure if, because of the texture of the seabed here, whether that will happen or not. So this is a long-term, very scientific research project that they're doing that could impact kind of kelp forests and carbon stuff, like carbon capture stuff in the future, and be quite relevant. Because I mean, if it is the good carbon sink that we think it could be in these different settings, or if there's a way of doing that, then this can obviously really help with the sort of the climate change targets that all the governments are doing. So we could see like kelp rewilding projects all over, you know, and things like that. So yeah, it was fascinating to learn all that. And I mean, there was, I'd sort of mistakenly understood that there was a bit of controversy around that, but it's different areas and different bits that the controversy is more around. So that's all described on the blog in this conversation as well. We really go into detail on all that. So yeah,
Brilliant.
it's fascinating.
Brilliant. I mean, there is so much information on the blog. It feels like you've only just begun the process, and you intimate that on your last blog post, that you know you've got some quite strong kind of leads in terms of where you want to take the research into work. Do you want to talk a little bit more about that?
I mean, one thing that I've discovered part of the way through the research, I stumbled upon, I think it's called a Google whack. It's where you Google something, you get one hit, one thing comes up. I was looking for British seaweed agar, I think, or microbiology as well, maybe like those words. And I found an article in the Microbiologist magazine that Stephen Forsyth, who I interview on the blog, I tracked him down and interview him on the blog, had published this article because his great uncle had worked on a project in World War II, where they'd had Sea Scouts and stuff hunting for British seaweeds that could make agar. Because I've already mentioned penicillin and the development of penicillin, which was in the Second World War. We needed agar in the Second World War to grow bacteria to do these experiments on. We needed a lot of it. And all the agar came from the Far East, and that was really, really problematic in this time. So So the fascinating thing was this discovery of this. It was called a short report into the utilisation of certain British seaweeds in the production of agar. So I got that, and I tried to replicate some of the experiments in making my own agar. But then when I actually interviewed Steve, he put me onto this book, which I managed to get a hold of a copy of, and I spent some of the budget on. That's okay.
Yeah, definitely.
And it's called a study of certain British seaweed. So the short report was just like an abridged version of this gorgeous book that has all these wonderful kind of images and maps and all sorts of things in it. So it's very rich on all the different seaweeds that they tested. And it's got a review of like all the global agar seaweed options that they use in different countries and things like that as well. And I didn't have time to do anything like a scientific study into it. I just kind of did a sort of boiling up seaweed in the kitchen. I mean, there's lots of different methods you can. There's some seaweeds that are better if you add vinegar to them at a certain point. There's some seaweeds that, I discovered one thing about how freezing can remove the non-gelling agents. So I froze the petri dishes that I made and then reboiled it and tried to regrow it.
Did it work? Did it work?
Do you know they're on the floor? I could go and get them. They're over there, and we can have a look, see what I grew. Hang on, I'll do it. Mind. Nobody watching on mine. So let's have a look. Here's one I made earlier. Oh, you can see. So I'm growing it on silk. I don't know how well you can.
Wow.
You can see it. I've got a light.
Yeah, I can see that really well.
And you see that texture of the bacteria growing on it there.
Yeah.
Different texture there.
That's definitely set though, isn't it?
It's definitely set. And then I embedded some silk in it, because one thing I tend to do is grow bacteria onto cloth. So it's a layer of agar, then the silk that's botanically printed with the things, with the seaweed, which is looks like a bladder rack, because you can see the bladder on it, and then you can see that glistening. You see that sort of glistening where it's sort of wobbly.
Yeah, really see that well.
Bacteria growing. So
right.
Yeah, I've got a few of those.
So where is this this process going, Anna? What do you want to do next with it to delve more into agar and seaweed with the big tome that you just showed us?
Yeah, I mean, I would love to kind of just look into it more with some microbiologists or seaweed experts or phycologists and that sort of thing, and see if we could kind of develop something like that. It would be really interesting to know if there were good alternatives that we could kind of forage, especially for this, like this Sargassum muticum, for instance. I think it makes alginate. I don't think it makes agar as such, but I mean, there are all these different things. So there's agarose, alginate, agar, all sorts of different things, and there's something else beginning with C. I can't remember the name of it. That these seaweeds produce that are kind of jellies. And the one thing, so seaweed seems to be, compared to something like microbiology, hugely under-researched. And I think usually when things are under-researched, it's because of money and lack of tools. So with microbiology nowadays, we have this whole genome sequencing stuff. It's like a gold rush to discover a new thing. And you can literally, if you read the microbiology like tweets and stuff like that, and the news stories, there's a new discovery, like a major new discovery almost every day in microbiology nowadays, because we have these new tools like whole genome sequencing, and especially things like nanopore whole genome sequencing, where you've got these kind of tiny like USB whole genome sequencers and stuff like that. And obviously you can sequence the genomes of seaweed, but compared to bacteria, it's the tip of the iceberg of sequencing. So we know very little about the genes that they have and things like that. And then I'm very interested in things like synthetic biology, which is like, you know, genetic modification of things, usually doing knockouts. With plants, they usually knock out genes to make them, like, things that interfere with the production of something you want it to produce. Sometimes they have these, like plants, for instance, will have protection things that stop making them as useful, and so you can kind of reduce the amount they protect themselves and protect them more yourself or something, and then have them produce other things. And so I'm interested in, like, what potential for synthetic biology is there in seaweed as well, and things like whole genome sequencing. There doesn't seem to be that many people working on it in the UK, which apparently has one of the highest numbers of seaweed diversity in the world, bizarrely. So I mean, the other thing I would like is, I'd like to maybe we can have another seaweed obsessed age, because there's this whole, and I was talking to Ella Garrod again, she was saying that they do a kind of citizen science thing on the beach, but during the Victorian era it was a craze. It was like people were mad on seaweed collecting and studying seaweed, and maybe these sort of things could come back again. It's something like, people in the coast can't travel as much at the moment and things. It could be something that we could do and we could kind of look at together.
Yeah, sounds like a research project, doesn't it, Anna? That's great. That's really brilliant. It sounds like you're going to be experimenting into the future.
Yeah.
Yeah,
it's just a start. Starting it as a sort of as a more in-depth project, because before that it was just it was me and my sort of interests and kind of a bit dabbly, I guess, on the fringes of my existing work. But now it's kind of kickstarted it as a much more in-depth research direction.
That's brilliant, and as is your blog, and it will be there for, I don't know, maybe forever. Thanks very much, Anna. It's been fantastic. Your enthusiasm as well as your hard work on this project.
Well, I should say that this algologies thing, it's a fusion between, and it's play on this idea of the multiple meanings of it, because also the idea of algology as a study of pain, it's under the shadow, the whole project's under the shadow of the COVID pandemic and everything happening in everybody's lives at that time as well. So there's sort of that side to it as well, which I think comes through more in the blog than my sort of kind of crazy biohacking enthusiasm. But anyway,
yeah, no, it's great. It's great to have that poetic comment, actually, well, both at the beginning and at the end of this conversation. Yeah, we wouldn't expect anything less from an artist. Thanks, Anna.
Thank you, Liz. Bye.
Bye.